Hello, class. I hope you're doing well. In this module, we are going to start chapter 3, which is going to be on cells, and we will finish chapter 3 up next week. So, to start off with, we want to think about cells, that they are the basic unit of life. And so, whenever we discuss the idea of a cell, you have to think about it as the basic unit and structure and function of the body. So, we've talked now at this point from a chemical level, and now we're moving on to the cellular level. And when we think about cells, they're very, very tiny. We have thousands to millions of cells that are making up our bodies today, and they're measured in micrometers. So when we think about micrometers, they are extremely small. And usually we show it as this funny little U and the M to kind of go with it. So again, very, very tiny little guys. And what's so cool about our cells is we have multiple different types of cells. They're specialized and they have certain characteristics about them because, again, when we think about structure, it relates to their function. So, when we discuss the idea of having specialized cells, we use the term differentiated. So, differentiated cells tell you that they're very specialized in a particular function. So again, we have hair cells that are specialized. We have nervous system cells that are specialized. We have muscle cells that are specialized. So we use the term differentiated. And another thing to keep in mind as well is although we have several different types of cells, they do vary in size and shape because remember, their structure is going to relate to their function. How they look is going to basically determine how they play a role within our bodies. So this is just to give you an idea of what to expect in terms of cells. So in letter A, I know it's kind of hard to tell, you're going to see a red blood cell. Red blood cells function by helping us to carry oxygen and gases throughout our tissues, primarily oxygen. And again, it's very, very tiny. This is 7.5 micrometers wide. So very tiny little cell. You have to actually utilize a microscope in order to see them. In figure B, we have a white blood cell. And when we discuss white blood cells in particular, they're going to help with immune system function, as well as some of them can help with inflammation or what's called phagocytosis, where they actually do a little bit of cell eating. And again, quite small, they're about 12 micrometers big, and you would have to utilize a microscope to see them. Now, moving on to figure C and D, we can see how, again, we have a big variation in size. We go from 7.5 micrometers to 140 micrometers. And then in figure D, we can see 200 micrometers. As you notice, their shapes aren't the same either because, again, the shape is going to determine their function. So like over here, when we look at figure A, this is showing you a nervous system cell. And it has these long extensions that help with sending electrical impulse. So their shape is really going to show you how they operate and what they do within our body cells. In figure B, we're going to see epithelial cells, and these are going to help to actually create underlying protection. And then finally, in figure C, we see cardiac cells. And these are very important to actually help with contraction in muscle cells as well. All right, so now when we think about what truly makes up a cell, there's not a typical cell, but we can think about sort of this composite cell, if you will, that shows you most of the components that you will find within a typical cell. Although, again, there's no such thing as a typical cell, but most of our cells have these particular functions. And there are three major parts to every cell. Every cell has to have these parts when we are discussing eukaryotic cells or animal cells. So our cells are going to contain a nucleus, a cytoplasm, and a cell membrane. And so what happens here, if I'm to draw a cell, imagine that this is your cell. And this thing in blue is going to be our cell membrane. This cell membrane is very important because it actually creates a barrier between the inside and the outside of the cell. On the inside, we're going to see that we have a cytoplasm. And the cytoplasm is going to be full of a liquid component called cytosol. But we're also going to find particular organelles within this region as well. So I'm just going to show them as this point in time. Imagine that that's our organelles. But the cytoplasm is the medium in which, again, we will find our organelles and we'll find the liquid cytosol. And then another major part of a human cell, or what we call animal cell, is a nucleus. And the nucleus, I'm going to show in purple, is a big, huge ball, usually in the center of the cell. This is where we're going to contain DNA. DNA is our genetic material. And if you need to go back and review, again, DNA is deoxyribonucleic acid. It's our genetic material, our genetic code, and it is double-stranded, and it's made up of nucleotides. But what this genetic material does is it directs cellular activities. So we have to have a nucleus, we have to have a cytoplasm, and we have to have the cell membrane to kind of create a barrier. All right, so here is a really typical picture that you'll see in textbooks of an animal cell. So as you will see, there is a lot going on within the inside of the cell, but there's also quite a bit going on the outside of the cell. On the outside of the cell, again, we have to keep in mind that we're going to have the cell membrane. And this cell membrane is what we see here. Now, if we were to magnify this picture just a little bit, we can actually see that the cell membrane is composed of what we call a phospholipid bilayer. So again, just a little bit of a review. We talked about phospholipids when we mentioned chemistry. Phospholipids are a type of macromolecule. They're an organic macromolecule, so they contain carbon and hydrogen. And it's made up of a glycerol head, a phosphate component, and 2 fatty acid tails. So the glycerol heads like to interact with water, and the tails do not, which is why they're going to be found in the middle. They're sort of like sandwiched together. And it creates what we call the phospholipid bilayer. We'll talk a little bit more about that here in just a moment. Now, again, on the inside of the cell, we are going to be able to find our cytoplasm. And our cytoplasm is going to contain all of these organelles, as well as a liquid suspension called cytosol. And then finally, we see our nucleus. And our nucleus is what's going to contain our DNA, that genetic material. Now, there are several other organelles going on in this picture, and we're going to talk about them one by one. But for right now, again, remember that every animal cell has those 3 components. Nucleus, they have to have a cell membrane, and they're going to have a cytoplasm. If they don't have those 3 components, they're not technically a cell. All right, so the cell membrane sometimes is also called the plasma membrane. So I put that in parentheses. Depending on your text, you may see, or they can actually, even in text, use them interchangeably, cell or plasma membrane. Again, that is going to be what creates this boundary around the cell. So there's going to be an inside and an outside. And it helps to maintain the integrity of the cell. But what it does is it separates the intracellular fluid, the inside portion, from the extracellular fluid, the outside portion. And we always say that it is selectively permeable. Selectively permeable means that it doesn't allow for everything to go in and everything to go out. Only certain things can enter and only certain things can exit. Now, it does utilize a communication called signal transduction, and it allows the cell to receive and respond to messages, because believe it or not, your cells have to talk to each other and have communication. And so this allows messages to go back and forth between cells. And what we're going to see is that the cell membrane is mostly composed of lipids, specifically phospholipids. We also will see a little bit of steroids. And we're going to see proteins. There are a handful of carbohydrates thrown in there as well, but primarily we'll see lots of lipids and proteins that are going to make up the cell membrane. And like I said before, the cell membrane is composed of what we call a phospholipid bilayer. So the phospholipid bilayer, again, thinking about phospholipids, they have that glycerol head, and then they have those fatty acid tails. The glycerol head is hydrophilic, meaning that it loves water. Philic means to love. Hydro is thinking of water. So the heads form the surfaces that are going to come into contact with your solution, your aqueous solution, or AKA your water. And the tails do not like water. They are hydrophobic. So those hydrophobic regions that we see within the tails do not want to come near water. So they form the interior portion. And that's why this is a bilayer, because you have to have two layers to basically hide in those tails. And the bilayer is permeable to lipid soluble substances. So if something is lipid soluble, it will be able to go straight through the bilayer. But if something is water soluble, it's not going to be able to just pass its way through. This is why when we talk about the cell membrane, we say that it is selectively permeable. It allows for some things to go straight through, but not others. Now, within this nice little lipid bilayer, we'll also find some cholesterol. And remember, cholesterol is also a lipid. It's a particular steroid. And imagine, if you will, that I'm just going to show it in yellow. We have it here to help to stabilize the membrane. It helps with a little bit of integrity, and it's going to help to keep, again, impermeable to water substances out. Now, we also are going to have proteins thrown in as well. And the proteins I'm going to show in green. Some proteins are going to go all the way through, and some will just be on one side or the other. But this is what creates what we call the fluid mosaic model. There's a mixture of these phospholipids, this bilayer, cholesterol, and some proteins. And some lipids and proteins can move, which is why they call it the fluid mosaic. Now, there's other types of membranes found within here. There's pores, there's channels, and there are some that help with what's called identification and cell adhesion molecules. We're not going to talk specifically about CAMs, so you don't have to worry about it for this class, but there's lots of different proteins that are found within this phospholipid bilayer as well. And then usually what we'll see on the surface is we're going to see some carbohydrates. And I'm going to kind of show it off like this in pink. And this is going to help with cell recognition, as well as helping with interaction and identification. So this is my terrible picture of the cell membrane, but the next picture that you'll see, it'll make a little bit more sense. So here again, if we were to kind of magnify this picture, we'd be able to see that this is the inside of the cell, the intracellular fluid, and this is the outside of the cell. And it's maintained as an inside and outside because of this cell membrane. This cell membrane is made up of, again, these polar or hydrophilic heads that want to interact with water. And then in the middle are these non-polar hydrophobic tails. And again, the tails do not want to interact with water. So because of this, it is selectively permeable. Those that are lipid soluble will be able to go straight in, versus those that are water soluble are not going to be able to because of these tails. So again, it only allows for passage of certain things in and out of the cell. And this is why we have proteins and channels and other mechanisms to get those substances within the cell. But it is very much regulated. And again, that's why we talk about the cell membrane being selectively permeable. All right, so here's a much better picture that I was trying to depict in my drawing. I apologize, I am a terrible artist, so I hope this makes a little bit more sense. But when you're going to look at the cell membrane, again, we can see the phospholipid bilayer. That's what we see in this portion here. We're also going to see different types of proteins. Again, some proteins are going to go straight through one side to the other. Some are just going to hang out on one side or the other. The proteins that go straight through from one side to the other are called integral proteins. The ones that stay on one side or the other are called peripheral proteins. And so they work in different manners. Some of them are receptors, some of them are channels, some of them are pores. And so they're going to have different functions within the cell. But a lot of them are really helpful for creating channels. And again, some of them are helpful for being markers or receptors. Now, like I said, we also have cholesterol thrown in there. And so we can see the cholesterol in yellow. And this is to help to maintain the integrity of the cell membrane. And again, to make sure that water-soluble molecules don't try to pass through. As well as, again, we can see these carbohydrates, these sugars that are on usually the outer surface, and they're going to help with recognition and communication. So this is the extracellular fluid. This is the outside of the cell. This is the intracellular fluid. This is the inside of the cell. All right, so to make sure we feel comfortable with some of those proteins, again, we can see different types. We have receptors that help to respond to signals from the outside. Some of them can be channels or pores, so they can help to actually transmit some of those molecules that normally wouldn't be able to pass through that cell membrane. We also have enzymes as well, and we're going to discuss enzymes in way more detail coming up. But enzymes are a particular protein that help to actually speed up chemical reactions. They catalyze or they speed up chemical reactions by lowering what's called the activation energy. We have those surface cell proteins that help to establish their self, so that way our cells can recognize it as something that is self and not foreign. And then again, we have those CAMs that help them to stick to each other. We will not be talking about CAMs in detail at this point, so you don't have to worry about that. All right, so really quickly, I like to make mention of some clinical applications. So sometimes you'll see issues with ion channels. And ion channels are protein channels that help to carry sodium and potassium. Sometimes we can also see chloride channels being used as well. And what they're going to do is they're going to move electrolytes from one side of the cell to the other, so extracellular to intracellular, or intracellular to extracellular. But what we can see is that faulty ion channels can actually cause some diseases. So there's mutations in some channels, like sodium channels, that cause the inability to feel pain or extreme pain conditions. So if you do not have proper sodium channels and there's this sort of leaking, if you will, it can cause the inability to feel pain. If there's mutations in potassium channels, it can actually disrupt electrical activity. And a lot of times we can see this led to cardiovascular issues, such as problems with heart rhythm, as well as impaired hearing as well. And then abnormal chloride channels can actually cause cystic fibrosis. And cystic fibrosis is a genetic disorder that usually causes a production of a very thick mucus. So it can lead to issues in respiratory, in your pancreas, as well as it can lead to reproduction issues. And usually one manifestation of someone having cystic fibrosis is they have very salty sweat. All right. So we talked about the idea of the cell membrane. Let's go ahead and talk about the cytoplasm. The cytoplasm is going to be the inside of the cell now. And what we're going to see is that we have our membranes and organelles, and they're suspended in this liquid substance called cytosol. And cytosol is the fluid portion that we're going to see, as well as we'll also notice those organelles. Organelles are tiny little structures with specific functions. So imagine, if you will, that they're sort of like baby cells that have a very specific function in mind, and they help to carry out the cell's activities. So they're all little workhorses that are keeping the cell basically alive and regulated. And those organelles are extremely important. If you have faulty issues with organelles, you may actually see problems at a tissue level, organ level, and even an organ system level. And what we'll also notice too is that there's this cytoskeleton as well that kind of provides a framework to the cell. And so this is where we're going to find some supporting frameworks like rods and tubules and microtubules found within. Now, there are several different organelles found in the cytoplasm. One of which that we'll talk about are ribosomes. Now, ribosomes can either be free or they can be bound to something called the rough endoplasmic reticulum, or the RER. So imagine in the cell that you have these little dots, if you will, and they can be free or they can be bound to something called the endoplasmic reticulum. And then if they are found on here, they're called the rough ER, or rough endoplasmic reticulum. And they're going to help to provide structural support, as well as they're going to also aid in making amino acids for protein synthesis. Again, proteins are essential to our bodies. They are going to be a very important macromolecule because they help to make hormones, antibodies, enzymes. And in order to make proteins, again, you have to have amino acids. That's the building block. So the ribosomes are going to help to link amino acids and help with that protein synthesis to actually make them. We have the endoplasmic reticulum, and there's two components. There's the rough ER that has a sort of bumpy look to it, and then there is something called the smooth ER. And the smooth ER does not have those ribosomes attached to them. But what you're going to see is they're like membrane-bound sacs that are kind of all together, and they're making up this tubular-like system. So the rough ER contains ribosomes, so it helps with protein synthesis. The smooth ER, of which I'm kind of trying to depict in green, does not have ribosomes, and it's smooth like butter, kind of the way to remember it. It's going to help to conduct lipid synthesis or fat synthesis. All right, so here's a much better picture to see in detail. So when you're looking at these membranes, again, we can see the rough ER and we can see the smooth ER. The rough ER has a rough-like appearance to it because it contains those ribosomes. Those ribosomes are those little dots. And like I said, ribosomes can also just be freely found within the cell as well, but they can be bound to the rough ER. And they're going to help with protein synthesis. Versus the smooth ER has no rough-like appearance to it. It's smooth like butter. And this one's going to aid in lipid or fat synthesis. All right, we have some other organelles as well that we'll discuss. So we can have vesicles, which are little sacs that can help to store or transport substances in and out of the cell. So imagine, if you will, that there's sort of like a little bubble, if you will. And so it's what we call a membranous sac. And again, it can help to store substances as well as transport them out of the cell if we need to. We also have what's known as the Golgi apparatus. This one sort of looks like a stack of flattened pancakes when you see it. But what this does is it picks up the proteins that were made from the rough ER, and it double checks them like the post office, if you will. So whenever I discuss the Golgi, I kind of call it the post office. It's going to help to refine, and it's going to make sure that the protein is made correctly. And if everything is good and ready to go, it'll package it and deliver the protein wherever it needs to go. And that will butt off in terms of a vesicle. Now, we can also see that there is a smooth ER component as well. And we can actually have milk secretion as an example among the rough ER and smooth ER, as well as, again, once we have that sort of refining and packaging of the Golgi, it can be sent out via those transport vesicles. All right, so here's a nice little picture of the Golgi apparatus. Like I said, I look at it and see a stack of flattened pancakes, and then you can see these little vesicles kind of popping off of it right here. So a very important little component. And then again, this would be our Golgi apparatus that we are able to see. All right, we also have a mitochondria, and the mitochondria is a membrane-bound, fluid-filled sac. Kind of looks like this when we see it. It has these inner and outer membrane folds. But this guy is known as the powerhouse of the cell. And the reason we describe it as the powerhouse is it's going to actually allow for cellular respiration to take place. And this is going to help to produce energy. The energy in which we use here is called ATP. That's the cellular energy that we need in order to do work. So the mitochondria actually helps with that ATP production. And that's why we call it the powerhouse of the cell. We're also going to see that we have lysosomes and peroxisomes. They are difficult to distinguish when you're looking at the cell. So I wouldn't necessarily ask you to identify one or the other, or if I did, I would give a little bit more detail to it, because just looking at the overall picture, they look pretty similar. They're both going to be sacs, kind of like a vesicle, but the lysosomes actually contain digestive enzymes that are going to help to break down proteins and carbohydrates. On top of that, they also help to break down anything that's excess, as well as debris, worn out cell parts, and even bacteria as well. They have these digestive enzymes within them that they sort of work like a garbage disposal. And then we have peroxisomes, which again are very similar to lysosomes, but they're going to really help with the digestion of lipids, alcohols, and hydrogen peroxide. So these components that if we were to leave them within our cell, they may actually cause toxicity or free radical buildup, and they help to break them down into something that's a little bit safer. All right, so that's what we can see here. We notice our mitochondria, our powerhouse of the cell. And again, that's going to help with that ATP production, as well as we can see those lysosomes. And again, within the lysosomes contain their digestive enzymes to break down worn down parts, excessive components, and even bacteria as well. All right, we have some cytoplasmic structures as well that are going to help to create the cytoskeleton. I won't ask you this in detail, but there are microfilaments that are going to be made up of rods. We have microtubules as well. And then we have some intermediate filaments as well. All I want you to know is that these guys just create the cytoskeleton, and they are going to be part of kind of maintaining the framework of the cell. So they're going to help with moving organelles. They're going to help with muscle contraction. And again, they help to provide that cytoskeleton. And that's what we can see within here. So all this little bridging taking place, all these guys down here, they're going to help aid in that cytoskeleton. All right, other structures that I would like you to know is like the centrosome. Sometimes it's also called a centriole. The centrosome is the actual body, and it consists of two centrioles kind of put together. And what you'll notice is that they're very close to the nucleus because they're going to help in cell division. We'll bring these guys back up here pretty quickly when we discuss the idea of the cell dividing and going through what's called mitosis. But they help to produce these spindle fibers, and they're going to help to actually form new cells. So we start with one parent cell, and we're able to replicate into two daughter cells utilizing these centrosomes. And to me, when you look at them, they sort of look like if you bit a piece of Twizzler. So if you guys have ever eaten the Twizzler candy and you took a bite into it, that's sort of what it reminds me of when you go to look at it. And like I said, it's made up of a centrosome, and then it has two centrioles that kind of come together, if you will. All right, we have some other structures as well that we're going to see externally, so on the outside of the cell. We have cilia, which are going to be like little hair-like fibers or fringe. Not all cells are going to have cilia. In fact, I actually have a video for you guys to watch if you go to the optional resources, and it discussed that some cells are going to really require the cilia, but not all of them. Usually we find it in the respiratory tract or we find it in the reproductive tract as well. So they form a fringe on the outside and they're very abundant. And what they do is they actually kind of create this beat back and forth in a very coordinated manner. So that's why we help it with propelling of mucus in the respiratory tract, as well as for females, it's going to help to propel the egg toward the uterus within the fallopian tubes. Now, a really long extension that we're going to see of the cell membrane is a tail. There is only one animal cell or one human cell that contains a tail, and that would be the sperm cell. Otherwise, other cells like bacteria and different types of fungi and different types of protozoa will have tails. We, on the other hand, only one animal cell is going to have it, and again, that would be your sperm cells. So the flagella is a very long extension, if you will, and it's going to help with motility so the cell can move back and forth. And again, if you think about a sperm cell, its whole goal is to swim towards the egg in order to cause that fertilization to take place. So it only has one flagellum. So instead of using flagella, we say flagellum, but there's only one known in the human body. So it would be the sperm cell. So make sure you know that. I will ask you that for sure. All right, so up at the top here, we can see those cilia, those hair-like fibers that sort of form a fringe. And like I said, they have a rhythm in which they move and they propel. So they help with that production and movement of mucus, as well as, again, we do see it within the fallopian tubes to help propel the egg towards the uterus. And then finally, again, we can see that flagella. Let me use a different color. We can see that flagella within the sperm cells to help with motility. All right, now we may also see some other little extensions on the outside of the cell. These are part of the cell membrane and they're called microvilli. Microvilli kind of look like little, almost like finger-like projections. And they're going to help to increase surface area when it comes to absorption. So when we talk about absorption of nutrients, especially like for our digestive system, we're going to utilize these microvilli. Imagine, if you will, that they're not hair-like components, more like finger-like projections that you're going to see. And then some cells may have what are called inclusion bodies. These are chemicals that are not necessary for survival, but they are utilized in different ways. So for instance, you may see like inclusions for melanin in the epidermis that helps with coloration, or you may see fat and fat cells as well that have these inclusion bodies. So they're only present in certain types of cells. And again, usually it's not necessary for survival. It just helps to aid in their performance, whatever that specialized or differentiated cell may be doing. All right, so at the top, again, you can see those finger-like projections called microvilli. Do not confuse them with fimbriae. Fimbriae are the hair-like projections that sort of form a fringe. Microvilli are going to be these little finger-like projections. All right, so like I said before, organelles are extremely important. And so from a practical application perspective, if you have diseases at the organelle level, you will see problems at an organ level, an organ system level, and again, they may even lead to death. So the first one that we see is this MELAS, which is a mutant gene of the mitochondria, actually. The mitochondrial DNA has a mutant gene. And the person can't extract the maximum amount of energy from their nutrients. We have this Krabbe disease, which is going to be an inability to produce one particular lysosome. And because of that, they can't produce the myelin for their nerve cells, and it leads to severe damage to their nervous system. We also have a disease called Tay-Sachs disease that does something similar, and it's due to an issue with the lysosome not being able to properly break down a particular ganglioside. And then we have ADL. This is caused by a lack of protein in the membrane of peroxisomes. So we actually have fatty acid buildup, and it can destroy myelin sheaths as well. So the nerve cells don't actually transmit fast enough, unfortunately. And again, we can see problems with the nervous system dysfunction. All right, and then finally, last but not least, we have to talk about the cell nucleus. So the nucleus is that big hunking ball in the middle of the cell. It's going to contain a nuclear envelope, and then within the nuclear envelope, we're going to find the nucleus. So imagine, if you will, that this guy was inside of the cell. You have a big old hunking ball, and then you have another ball within that ball. So the nucleus is going to be this portion here. And again, it's made up of a double layered membrane called the nuclear envelope. And then inside of the nucleus, we have the nucleolus. This is going to help with RNA production, as well as it's the site for making ribosomes. So a very important little component there. How we're able to make ribosomes is because of this nucleolus on the inside. But like I said, the nucleus actually contains our genetic material. So our DNA is found within here. And so we can see that in blue. And that helps to control the cell activities. And within this genetic material, we call it chromatin. This is the cell's chromosomes, which contain the DNA wrapped around these particular proteins. And again, it stores information for protein synthesis. Okay, so here we can see the nucleus. And again, we can see that nuclear envelope, which is going to be this double layered membrane. And within the nucleus, we find that nucleolus, which is that site for ribosome production. All right, guys, I'm hoping you're having a wonderful day. Go ahead and rewatch this video if you need to get more detail, as well as look at some of those optional resources as well to get better pictures and more 3D images of the organelles. Thank you so much, and I hope you have a wonderful day.